Ready when you are, Sabine.
Thank you so much. Welcome, everyone. I see people are slowly getting into this webinar. We have about 100 people registered, and I see there are at least another 55 to 60 online. So thank you all for joining this webinar. And maybe just give it one more minute to make sure everyone can join from the very beginning. I think we still have a couple people in the waiting room. Bernie, yeah, thank you. Great. Welcome, everyone. Welcome to this webinar today from ACTA STING, from a Statistics in Trials Interest Group from ACTA. Today we'll have Professor John Carlin presenting on tackling dichotomania and other statistical diseases in the pursuit of better science. My name is Sabina. I'm a biostatistician and I'm a member of the ACTA STING Executive Group, and I will be facilitating this webinar with John today. Before we start, we would like to begin by acknowledging the traditional custodians of the land on which we meet today and pay respects to elders past, present, and emerging. And we extend that respect to Aboriginal and Torres Strait Islander people here today. Just some welcome and housekeeping. So as you were joining this meeting, you probably noticed that your video and your microphone are automatically turned off. At the end of this session, we will have a Q&A, and then we can unmute you if you would like to ask a question. So in question time, please feel free to raise your hand or put a question in the chat, and then we'll follow up on that question together with John. A recording of the webinar will become available, and you can watch it on the ACTA website or on the ACTA YouTube channel. And we hope that you stick around until the end. So there is a post-webinar survey. We would appreciate your feedback on the webinar and also any other ideas you have for future webinars here with ACTA. So our speaker of today probably doesn't need a lot of introduction. And I recognize some names there of people attending this webinar. So you're probably all very well familiar with John. So Professor Carling is a Professor Emeritus in Biostatistics at the University of Melbourne and the Murdoch Children's Research Institute, where he was the head of Clinical Epidemiology and Biostatistics Unit for 20 years until not so long ago. And John is very well recognized for his leadership in biostatistics, and his career has featured extensive collaborative engagement in clinical trials and observational studies, both applied, but also in a methodological research on topics such as handling missing data, causal inference, and problems in the use of statistical methods. He was also elected as a fellow of the Australian Academy of Health and Medical Sciences in 2018. So the topic of today will be tackling dichotomania and other statistical diseases in pursuit of better science. In about 10 years ago, there was a statement by the American Statistical Association with recommendations against the dichotomization of the interpretation of p-values. So John will review the history of the tenacious grips of statistical significance on scientific thinking, aiming to better clarify the underlying problems and summarize progress towards better practice. Without much further ado, I would like to hand over to John now and let John start the webinar. So I'll stop sharing, and then John, feel free to share your slides when you're ready.
Thanks very much, Sabine, and it's great to see so many people here. I hope I'll have a few interesting things to say, perhaps a little controversial, but we like to be controversial, so that should be fun. Now, sharing my slides. Here we go. And you've seen the title already, so and the abstract there made it all sound rather ambitious. Of course, we won't be able to solve all the problems, but hopefully give you some things to think about and bring everybody a little bit up to date with this controversy, which hopefully many of you are aware of and have thought about to some extent at least. So here's my outline, a bit of the history, a bit of a review of the problem that we're talking about, then some new research on the recent literature, what's happened recently with this problem, and then some wrap up on where do we go from here? What can we do to make things better? So my abstract there began with this sentence: 10 years after the American Statistical Association released a statement recommending against the dichotomous interpretation of p-values, what has changed and what still needs to change? So I looked at this, and when I was starting on the slides, the words 10 years after stuck in my mind, ringing a bell with a record album that I once had and probably still have. There may not be any others here who can remember the late 60s and early 1970s. This was I'm not sure that Andrew Forbes is here. He would be the most likely. A rock band 10 years after. And amazingly enough, one of their songs was called I'd Love to Change the World. So that's a good theme for the talk and for a lot of my work, in fact. So 10 years since, we just changed that word from after to since. Here are the 6 points that this ASA statement, this rather famous statement now that was agreed to by the, I guess, whatever the committee of the American Statistical Association, not without some controversy. There was a lot of discussion around this. I was actually some part of a working group that eventually thrashed out these 6 points that could be agreed upon. P-values can indicate how incompatible the data are with a specified statistical model, but they do not measure the probability that the studied hypothesis is true or the probability that the data were produced by random chance alone. A very popular misinterpretation. Scientific conclusions and business or policy decisions should not be based only on whether a p-value passes a specific threshold. And 3 more points there that I can let you read for yourselves. By itself, a p-value does not provide a good measure of evidence regarding a model or a hypothesis. So 10 years since then, 8 years since a landmark paper that I was sort of honoured to be involved with, along with a number of rather more famous people like Sander Greenland and co. I recently checked on how this paper was faring and was a little bit shocked to see 4,357 citations. So this paper has had some impact, I think, and I can recommend it, of course. 7 years since Nature actually published quite a significant commentary, again involving Sander Greenland with a couple of others. And associated with this, there was an opportunity for scientists and statisticians to sign a sort of a petition calling for an end to the use of this concept of statistical significance. And Nature even published an editorial accompanying this commentary. This was a fairly brief commentary, but the editorial said it's time to talk about ditching statistical significance. They didn't really follow through on that editorial, but still, it's out there. It's 6 years since I last gave a talk on this topic, beyond dichotomous thinking in clinical trials. So this was given at the ISCB in 2020. I was very much looking forward to my first visit to Krakow in Poland. But instead I gave this talk from the front room of my house here in Melbourne at 2 a.m. So this was a little bit of a letdown, this invited session. I had high hopes that we would use it to to rustle up a new movement to carry this agenda forward, but it was a little difficult given the circumstances with COVID and so on. All right, so that's sort of 6, 7, 8, 9, 10 years ago. But this is not a new topic. There's a literature that goes back for decades, and I've just collected here a few representative titles spanning the history, choosing some titles that have more or less sort of catchy aspects to them. The Cult of Statistical Significance, there's a book-length treatment on the topic. This second reference here I'll come back to. I think that's a really useful article for our purposes in biostatistics. Significantitis, that's another term. I think this was perhaps the first time it was used, another new word to describe this particular form of dichotomania that we're talking about. A famous psychologist, psychometrician, Jacob Cohen, Gardner and Altman. This was a bit of a landmark in the 1980s when the British Medical Journal, the BMJ, took up the topic with a fair bit of enthusiasm that confidence intervals were really the important concept rather than p-values, estimation rather than hypothesis testing. So that idea goes back 40 years now, very centrally in biostatistics. 1970, another book, I think a collection of essays. 1960, The Fallacy of Null Hypothesis Significance Test. 1942, all the way back to 1919, when the ideas hadn't really even been properly formalized, but this far-sighted statistician with the unfortunate name of Edwin Boring wrote this short piece that really talks about some of the same issues that I'm touching on today. So what exactly is the problem that we're talking about? This dichotomania disease, if you like. What's the dichotomization of statistical conclusions, I guess, to put it simply. So I've got a few examples here. This one I've used a few times in the past, and it always seems to work. It just seems to encapsulate a lot of the issues very nicely, including the fact that it comes from the preeminent journal of medical research, the New England Journal of Medicine. It's a simple study, as many of their studies are, a randomized trial looking at the effect of simvastatin for acute respiratory distress syndrome. We don't really need to know much about the background. We can focus very effectively on one sentence in the results and one sentence in the conclusions from the abstract. The results say there was no significant difference between the study groups in the mean number of ventilator-free days. So that was clearly their primary outcome measure. 12.6 days plus or minus 9.9 with the new drug simvastatin, and 11.5 with placebo, p equals 0.21. And what did they conclude on the basis of those results? They concluded that simvastatin therapy, although safe and associated with minimal adverse effects, did not improve clinical outcomes. Okay, so this very bold statement, it did not improve clinical outcomes. That's the essence of what we mean by the dichotomania, that people make this statement. There was an effect, there wasn't an effect. So, you know, more formally they're thinking in terms of null hypothesis testing and the p-value being greater than the significance threshold. So we say the null hypothesis is not rejected, and then go to that conclusion that there being no effect. But hang on a minute. If we look at the data a little more carefully, we find in the text of the paper a discussion of sample size, of course. The sample size was 524 patients, on the basis that this would give the study 80% power at a two-tailed significance level 0.05 to detect a mean between group difference of 2.6 ventilator-free days. Okay, so that was their, if you like, their alternative hypothesis of interest. They didn't want to miss a difference that might be there, a mean difference of 2.6 days. So let's put that into context and look at the results in a little more detail in the form of the confidence interval and 95% confidence interval for the mean difference. You may recall the mean difference was 1.1 days ventilator free, but did the results in the abstract give the interval? No, you see the results in the abstract did not give the confidence interval for now that has changed over time. So you are more likely now to see in the abstract the confidence interval for the mean difference. And if that had been there, perhaps one might have seen the absurdity a little more clearly, because what we can see is that the effect size used for the sample size calculation is actually within the 95% confidence interval. So it's just about as consistent with the data as the null hypothesis of zero difference. So why are we saying in the abstract that there was no effect when which is zero, when we could just as logically say, well, there's an effect of 2.6. If we want to focus on a single point effect, we've given that zero incredible kind of priority in an illogical way. So how does this create a problem if it's not obvious? I mean, maybe my sort of the many years I've thought about this topic won't make me seem too dogmatic, but the absurdities just never seem to fail to amaze me. What should the reader take away from this paper? Well, not that the drug improves clinical outcomes, so we should use it. So we shouldn't take that conclusion away. That's the conclusion that has been presented, claimed, but we should equally not take away the conclusion that the drug has no effect on clinical. Sorry, this is the effect that's been claimed, that the drug has no effect on clinical outcomes. So we should forget about it. There's always this sort of implied kind of conclusion from this sort of reporting that this is the answer. So let's get on with it. We'll now use this drug, or we'll now forget about this drug. It's a very sort of black and white conclusion, which is clearly logically a false dichotomy, and it's clearly not going to be acted upon in that, in almost any situation. It's clearly not going to be acted on in that black and white dichotomous way. In fact, the only sensible conclusion here is that there was some uncertain evidence, potential benefit. We have an estimated effect, and we should move on from there and discuss whether we want to pursue further studies of this question to get a better estimate of the effect. It's complicated, right? Where do you go from there? But the consequences of dichotomizing is that you really leave the readership confused, and readers, stakeholders put in this situation because they've been led to expect something that can't really be provided. They've been led down this path of statistical tests as a sort of magic potion that tells you a certain, a definite answer. All right, another much smaller example, just quickly, because it's so, and again, kind of, well, it's more outrageous, this one. This is from a short article, Schmidt and Rothman, mistaken inference caused by reliance on and misinterpretation of a significance test. They were, I think the first author was a cardiologist, and noted notice this article in which authors, these Chow et al. Authors, claimed, as per the quotation here, a unique finding of the present study was that the use of selective COX-2 inhibitors was not associated with AF atrial fibrillation risk, except in patients with chronic renal or pulmonary disease. They presented, I guess, this confidence interval, perhaps a p-value as well. Yeah, I think there was a p-value which was above 0.05, as you can tell by the fact that the 95% confidence interval overlaps 1 on a ratio effect measure. So this was taken to mean there was no association, and was claimed to actually contradict this other study, Schmidt et al., which reported the same relative risk, in fact. So the two, and this is sort of rather over the top meta-analysis, combining these two results, and lo and behold, the overall estimate is the same as the two estimates with a narrower confidence interval. But I guess it's just emphasizing that far from being inconsistent or contradictory, this result was actually perfectly in support of the other paper. So this is an extreme example of what happens quite often, that a negative study is said to contradict a previous positive study, because one of them has p less than 0.05 and the other one has p greater than 0.05. All right, a couple of little anecdotes then to round up this sort of little history. A colleague who may or may not be on the call gave me this example where they had submitted a manuscript to JAMA reporting a primary result in terms of a risk ratio of 1.05 with a 95% confidence interval from 1.00 to 1.10. The editor didn't like that because it didn't make clear whether the confidence interval included the null value or not. So they were asked to provide the p-value to 3 decimal places, which you can now see here, and the confidence interval to an additional decimal place. So it became not statistically, and they had to report that as did not meet statistical significance. So as if this 3rd decimal place here has added some meaningful scientific understanding to that more concise initial report. Another delightful editorial correspondence in the reverse direction. The copy editor rounded results to 1 digit. So the rate ratio here was suggested to be reported as 0.7 with a confidence interval from 0.7 to 0.8. And the author requested, well, can we have the 2nd decimal place? And the response was, well, thank you, but yeah, it doesn't really make any difference because it doesn't change the statistical significance of the reported data. As such, these changes begs the question of whether such accuracy is truly necessary. All right. I used to try to get recent examples. I did pull an abstract from the latest issue of JAMA. And there's some little signs of improvement in reporting here. They have this rather cute style now of including these visual abstracts. So you can get the picture of the whole study quite quickly. For our purposes, I suppose the main interest is in the overall conclusion. This was about positioning of infants and their risk of infants with bronchiolitis and their the risk of escalation of care. So did they do better when they were positioned in a prone position or in a supine position? And the adjusted odds ratio is 0.66 with a wide confidence interval, a p-value of 0.09. And so they did report that the prone positioning did not significantly reduce escalation of care, but they added a nice caveat, but this study was not definitive and further research is warranted. So exactly what's been added by saying it did not significantly reduce is my question there, is well, why don't we just report the confidence interval and say the study was not very definitive. We have a fairly broad, but we hope informative, broad confidence interval, but we hope still an informative set of data, probably hard to come by, that gives some indication of a potential benefit, something like that. All right, so why is dichotomania a problem if it's not already obvious? Just to try and sum up some of the issues. So when people claim they have obtained a significant difference in their comparison of interest, it's really not clear what should follow from that claim. It's very rare that it actually flows directly into some actions. So there's a lot of confusion about, in my thinking anyway, about the purpose of presenting statistical analyses like that. Confusion along the lines of, well, it's like, now we know there's an effect, or now we know there isn't, therefore we should do something. Very rarely is there that, therefore we should. I mean, there'll be recommendations, but there won't be immediate uptake or change of practice on the basis of a single test result. So the main thing that results from a significant claim is a ticket for publication. So there's this incentivization, prioritization of statistically significant claims. And when there's that tendency to report comparisons that are statistically significant, preferentially report such comparisons, they will inevitably be related to inflated estimates of effects. So that selection process filters towards upwardly biased effects, or effects that are effect estimates that are biased away from the null hypothesis. The more important downside of the practice is associated with claims of non-significant differences, and the common conflation of that with there being no effect, which is clearly wrong, and many people sort of realize that, but it's still a source of much confusion, and can lead, and this is a focus of the nature piece, it can lead to erroneous downplaying of potentially important effects. So I think we need to look a little more closely at what these claims mean, even mean logically. So I'll come back to that, but I better keep moving here. A little bit of history. Where does it come from? It was interesting to see that in this report of what I think is regarded as the first published randomized trial in medicine, a trial of antibiotic treatment of tuberculosis in the 1940s. Quite a small study, as you can see. 4 of 55 patients on the streptomycin therapy, 7%, and 14 of those on the control treatment, 27%, died before the end of 6 months. The difference between the two series is statistically significant. The probability of it occurring by chance is less than 1 in 100. So the p-value's not been quoted precisely, but it's buried in those words. But also in those words is one of the classic misinterpretations, this idea that the p-value is somehow the probability of the observed difference occurring by chance, or the probability of the difference that we observe being due to chance, which turns the logic of the p-value on its head, as I will probably say a little bit more about that. Why this dichotomy? I would like to know a bit more about this. It's quite hard to dig up. I think this book is probably quite an interesting read. I haven't read it yet, but I've read quotes from it. Surrogate science, the idol of a universal method for scientific inference. So I think it's about along the lines that scientists really would like to have this guide to truth. And when in the early part of the 20th century, statisticians produced these theories about testing hypotheses, it seemed to fit with the prevailing philosophy of science and was taken up very enthusiastically. This idea of like, oh, right, now we can test our hypotheses. The statisticians have figured out how to do this. And I think ever since it's been this sort of rather mysterious technical trick that, you know, you go to the statistician to get to because they know the right test, and then it's like, whoa, then the answer tells you whether you've found something, found some truth or not. I'm putting in a few comments more specifically about trials for this group. Hypothesis testing is especially strongly embedded, of course, in clinical trials. And I think that's partly linked to regulatory decision making, embedded in which there's often this step of, well, you can take your drug to the next stage of approval if you can show us a trial result that is statistically significant. So there is a decision point there. So it's convenient for the regulators, and in some ways then it just imposes itself on the science in what may be an unfortunate way. Outside of that area of regulatory decision making, however, there doesn't even seem to be that sort of case for significance testing, because, as I said before, most trials don't lead directly to an actual decision. And when they do, it's not usually driven purely by one hypothesis test result. All right, so looking a bit more closely at the dichotomy, I just wanted to flesh out a little bit the logical confusion that's involved. There were two great sort of traditions of statistical testing that emerged in this period, when, as I mentioned, there seemed to be a lot of emphasis on finding ways to draw dichotomous conclusions, and Neyman and Pearson especially took this up, although they were well aware of the limitations of their theory with respect to drawing inferences from specific data sets. So the Neyman-Pearson theory is the theory around type 1 error, type 2 error, that we should conduct our statistical analyses, our experiments, and the analysis and reporting of them in such a way that over the long run, we don't make too many errors. And everybody here should be familiar with the two types of error, the type 1 error, the type of error that you make in the hypothetical universe where your null hypothesis is true, and the type 2 errors are the ones you make in the hypothetical world where the null hypothesis is false and some alternative is true. So that whole beautiful theory can be used for various purposes, and they spelled it out here in summary as a way of creating rules to govern our behavior with regard to which in the long run of experience we shall not often be wrong. So they were not, they were explicitly, and they were well aware of the fact that they were not claiming to provide a method of interpreting any particular specific data set. It was just like, here, do things like this, and then over time you'll be pretty good at drawing good conclusions. Fisher was much more sort of empirically based. He wanted to say things about data that they collected in agricultural experiments and so on. So he emphasized the p-value and talked about how you could consider it as a more or less continuous indicator indicating surprise, that if this p-value thing is small, then you've observed something that would be surprising if there was no difference in the effect in the true world that you can never see, in the infinite sample size that you can never collect. So what happened was that these two traditions just got mashed up together, and this is very nicely spelled out in that paper of Stephen Goodman's that I had in the earlier reference list towards evidence-based medical statistics, part 1, the p-value fallacy. So the standard practice that we see all around us is that people define their test statistic, calculate their p-value, which is the probability that that test statistic would have been under random variation in the universe where the null hypothesis is true, the probability that that test statistic would have been greater than the actual one we've observed, T-obs. And if that p-value is small, then we declare less than a threshold, then we declare the statistical significance has been observed. So that's claiming a scientific inference from the p-value. It's saying, well, given this data set and this calculated value of T-obs in the data, I'm going to claim something about the data set. I'm going to claim whatever it means, that H naught has been rejected or disproven. But it doesn't make sense. There's no logical way in which the fact that this p-value is less than an arbitrary threshold provides a logic for the conclusion of rejection or disproof of this hypothesis, because it's confusing these type 1 and 2 error rates, which don't apply to your specific data set here. They apply to the long run of analyses that you might do. It confuses those ideas with this actual decision based on so-called decision or conclusion based on the data set in front of you. And then from all that confusion, we get all the misinterpretation in both directions that I touched on before. So again, are trials different? Is there a special case for hypothesis tests in trials? There are stricter rules generally applied in trials. So there's more discipline than in many areas of medical and other quantitative research. But I would claim it's still fundamentally illogical and misleading, as per the example earlier. And that trial report shouldn't confuse the reporting of estimated effects with conclusions or decisions about recommended practices. They're two separate things. One feeds into the other, but they're not the same. And then the other thing that's often raised by trialists, well, don't we need this type 1 and type 2 error? Otherwise, sort of all our trials will kind of, there'll be the sort of wild west of people reporting all kinds of things. Well, it clearly does maintain a certain order to the rules around what we can and can't report. But if we moved away from the notion that individual trials have the job of reporting a conclusion about whether a treatment is or is not zero, if we moved away from that, then this wouldn't be so relevant at all. And I claim that this machinery is really just a game that's maintained in order to provide a framework for determining sample sizes, which is perhaps fair enough, but it would be nice if it was recognized as such. This is just the game we have to play. Was like, oh, well, let's put us in that mode of accept, reject hypotheses just for the purpose of figuring out a sample size. Still leaves you with the logical problem afterwards of, oh, now I've got to reject, or now I've got to accept, and what does that mean? All right, so there's the summary, and my exhortation is that statisticians shouldn't perpetuate this practice of drawing falsely dichotomous conclusions. So have things changed longer term and recently? What do we see in the journals? I did a quick look into these major journals, into their guidelines to authors, and was pretty disappointed, actually, to summarize quickly. I've got a few extracts here from JAMA. Avoid solely reporting the results of statistical hypothesis testing. So there's definitely some movement there, such as p-values. For most studies, p-values should follow the reporting of comparisons of absolute numbers or rates and measures of uncertainty. So they're saying it sounds as though they're going along the right lines, but comparison of absolute numbers or rates is a very confusing way of talking about effect sizes or treatment differences or meaningful parameters. So that's an important point that I want to come back to at the end. So I need to make sure I get there. Further in their guidelines, they use the word significance 4 times in what just ends up being kind of confusing, I would say. So let's skip through this fairly quickly so we don't run out of time. The New England Journal's situation is more complicated and more confusing, arguably. They are concerned about the adjective significant, and they want it only to mean statistically significant. They do say that effect estimates should be provided with standard errors or confidence intervals. But then they go into multiplicity considerations, and there's 4 lengthy paragraphs there that in my and others' minds constitute digging a big hole for everybody. So the method section should state that the widths of the intervals have not been adjusted for multiplicity. The intervals may not be used in place of hypothesis testing, as if we were going to do that anyway. The whole point of intervals is to get away from hypothesis testing. And then the interpretation of these confidence intervals should avoid the language of definitive conclusions used to report statistically significant findings as assessed by form. Anyway, so kind of moving away but not letting go is maybe one way of summarizing it, and just leaving everybody confused. The BMJ, please do not use the term negative to describe studies that have not found statistically significant differences. There'll always be some uncertainty. So that's all good. Then there are some other words here. Using words such as our results are compatible with a decrease of this much or an increase of this much, or this study found no effect is more accurate and helpful to readers than there was no effect, no difference, possibly. All right, so what do we see in some sort of empirical reviews of what's going out there in the major journals? This, I think, is one of the more interesting and comprehensive reviews that's been done. It's now a little bit out of date, so it's not able to tell us if anything's changed in the last 10 years. There's a bit of evidence on that in the next slide. But this one was interesting because it covered not only 5 major medical journals, but 7 epidemiology journals. And these are notable because several of them, most famously the journal Epidemiology, have had very strong editorial positions against the reporting of dichotomous hypothesis tests. And you can see that in the results that they report in this review. Overall, they report encouragingly that the CI-only approach, so that's where people report estimates of effects with confidence intervals, but not any statement of significance or p-value at all. But on the other hand, the hybrid approach, where people report the p-value and they also report significant, not significant, effect, no effect dichotomy, retain dominance in 4 of these 5 medical journals, though not in the epi journals. And yeah, they still very common in widely read medical journals, especially in the hybrid form that I just described. P-values are reported numerically along with declarations of the presence or absence of statistical significance. Another major review, which is more recent, goes up to 2025, by a group that published a similar review 10 or 15 years ago, in the group of John Ioannidis. They reported a rather less encouraging outlook, really, that the proportions of articles that report p-values has actually increased, so the proportion of articles reporting p-values increased from 7.5% in 1990 to 18% in 2025. And as I've highlighted here, the proportion that reported at least one significant p-value remained in the range 94 to 98%, which is really quite strong prima facie evidence of that selection process, that it's the significant tests that get reported. So what do we make of that? And the proportion reporting at least one less than 0.005 has increased over time. Demonstrates the pervasive entrenchment of p-values. There's been reviews, some of which are quite recent, of other with other focuses. This guy Andreas Stang has a whole slew of articles. This one is specifically on randomized trials, and again it's a bit of a mix, but the red is a steady decline, but only as low as 77% in the number of papers, number of trial reports in which significance language was used. The blue charts an increase in confidence interval reporting, which is coming up little by little. This is the latest article. I popped this in because it's the most optimistic from the down with dichotomania agenda point of view. Shows the strongest evidence for changes in practice in HIV and AIDS journals. Although he makes the caveat that clear increase in reporting of confidence intervals doesn't necessarily mean that people are not subject to dichotomania, in the sense that they will often report a confidence interval dichotomously. Null value is in or outside my confidence interval, rather than focusing on all of the values within the confidence interval as being of potential interest. There's a little bit of fun here, really. These couple of slides. I noticed in the review I did that there was quite a lot of articles coming out over the last few years on this disease within a disease of reporting trends towards significance, or words to that effect. to describe almost significant differences, or in fact non-significant differences. And I thought, well, maybe that's at least flagging the fact that there's some sort of recognition of the illogicality of all of this, but none of these reports seem to be terribly self-aware of that, and they I wasn't sure that they were aware of this fabulous website which goes back to 2013 or 14 or something, where this guy assembled hundreds, if not thousands, of examples of text that people have used in papers where they would really have liked a test to be significant, but it wasn't. So they said it was closely approaching significance, closely approximating, did not quite achieve. It's hilarious when you look at the whole list. And he very nicely summarized the logic here, that if you're really worried about whether or not you've made the threshold, you're buying into that being a meaningful thing, and therefore you shouldn't be anguishing about the actual value of the p-value. You either made it or you didn't. But if you don't think the threshold makes any sense, then don't talk about it, and interpret the p on its own. So this is a nice way of highlighting that p-value paradox that I spoke about. Another very recent review, or a recent review, flagging some discomfort and potentially indicative of changes in the air. So some changes happening, but what needs to happen more broadly, we need to redouble these efforts. I think we need to really remove statistical significance from the lexicon, de-emphasize hypothesis testing. As an aside, it's not the p-value per se, so I won't dwell on this aside, but people think this is banging on about p-values. Well, p-values are kind of a bit odd and a bit hard to interpret, and maybe that's why people are drawn to using them against thresholds. But it's not the p-value itself, it's the threshold that's the real cause of the main issues. So I just wanted to spend a couple more minutes. I'm not sure when I was supposed to finish, but probably before now. Connecting to other misuses and misinterpretations of statistical methods. I think it's a classic example of what we might call a ritualistic or at least a habitual application of technique. And another example that I've spent quite a lot of time looking at over the last few years is the use of more advanced statistical methods, in particular the ubiquitous regression models. Like, I see, your outcome is binary, let me fit your logistic regression. Sort of reflexive behavior that I exhibited myself early in my career. We thought we were very clever in the last century when we knew how to fit logistic regressions. But a lot of this work amounting to methods producing answers in search of questions, rather than going back to the questions before we launch into the analysis planning, let alone the analysis. And the connection with the significantitis issue is this, that if we recommend estimation, not testing, are we being clear enough about what we are estimating beyond a statistically defined effect? So if we're still wrapping up our statistical advice in sort of jargon terms that are not really grounded in the original research question, perhaps that's part of the difficulty. So here, just a quick plug for this paper of ours that came out last year, which presents an overview of the many misuses and abuses of regression methods, all of which exhibiting a version of that problem, that the underlying research question hasn't been made clear. So here's the grand unifying solution, that if we encourage people to have absolute clarity of purpose, and this is in a sense fairly straightforward for trials, the question is always a causal question, and you need to work through the details of the outcome measure, the intervention, and so on, and various potential complexities around the conduct of the trial and following up and so on. But from the type of question flows the endpoint. What is the estimand? So we've heard a lot about estimands in trials recently. There's a broader concept of estimands here, really, that we should be specifying the target quantity before even thinking about analysis plans, whether in a trial or other causal inference context. And then, is it really of interest to ask the question, is the causal effect positive, i.e. non-null? Is zero such a privileged value that we should put so much focus on it, rather than being clear about our estimand, assessing whether data can in principle provide a valid estimate, and then pursuing methods for estimation. So following that roadmap, we don't see any place for testing hypotheses as such. Sharp focus on estimating a quantity of meaningful interest, and perhaps this would help to facilitate a stronger movement towards the estimation, not testing mantra, so that the things that the confidence intervals report are not just some sort of the numbers or the vague terms that were presented in those guidelines to authors that I summarized earlier, but they're actually meaningful, scientifically important quantities. All right, 3 p.m. Was that when we were supposed to finish? I leave you to read this amazing quote from John Tukey. Who would have thought he wanted to abolish the null hypothesis way back when? I'll stop there, Sabine. Thank you.
Yeah, thank you so much, John. Thank you so much for all those reflections on the p-values. I have to say, as a clinical trial statistician, I often provide p-values to clinical collaborators, and they're received in different ways by different medical disciplines and also by different journals. And I also find it very difficult to get people moving away from p-values. So I'm glad to see that I'm not the only one if I'm looking at the literature. We did run out of time in order for our Q&A. So I see there's maybe one lucky person who can ask a question. There's one person who put their hand up, so maybe we'll just allow that one. I'm not sure, Bernie, if you can unmute.
Ian.
Maybe I can unmute. Would you be able to unshare the screen?
Oh, sorry.
Yes, yeah. I saw Ian has put his hand up. Ian, would you like to? I think Bernie might be able to unmute.
I have clicked the button. Ian, are you able to unmute?
Yes,
can you hear me
it's
now?
working. Yeah.
Yes.
Oh, yeah. Yeah, thanks, John. Great talk, and I've got a lot of sympathy with your basic thesis. And I would just like to reiterate one point that you made towards the end there, which is that your basic criticisms are a criticism of thresholds, not of p-values per se. And I think even more broadly, they're not necessarily a criticism of frequentist statistics in a broad sense, which it's easy to conflate these criticisms with. So it's very much about thresholding rather than those other issues. I was sort of waiting for you to bring up Bayesian interpretation. So I'll ask you a question about that. What's your reaction to the increasing pervasiveness of dichotomania in Bayesian statistics? All of the Bayesian trials that are being designed now declare a threshold for the posterior probability, things like 99%, 97.5%. That's really just bringing dichotomania into the Bayesian world, where you would have expected that there was some sort of escape from the very criticisms that you're referring to. What's your reaction to that?
Yes, good. I expected that question, Ian, and I would have said a bit more about my Bayesian past and Bayesian inclinations if I'd had time. They have faded away a little bit. I mean, I still think Bayesian logic is far preferable to frequentist logic in terms of summarizing what we think. You know, if we could invest the meaning in a confidence interval, call it a credible interval, call it some other interval, but an interval that we can believe in, within which we believe the true answer lies. That's a Bayesian interpretation of confidence interval. And I think most people would really love to have that, rather than the convoluted interpretations of like, oh, well, if we did this study over and over, this way of doing it would have captured the interval 95% of the time. Therefore, we think this interval's quite a good one. And
but the dichotomania in Bayesian inference, though.
Yeah, the dichotomania. So moving on to that, I just want to slip in that little appeal to Bayesian logic. But yeah, it's been a bit disappointing. I agree with you. I think it's partly driven by this the necessities of trial planning, which still haven't found a way out of the type 1, type 2 error kind of logic. So even if you design a trial with you plan to use a Bayesian methods to analyze your trial, in order to have a plan for the sample size, as I understand it, you're pretty much forced to go through that same game of saying, well, we're going to have a rule that says the drug has worked, and that rule is now stated in terms of posterior probability the effect is above a certain level. But in order to satisfy you about the sample sizes, we're going to have to do all these simulations to get some idea of how often, when we do this trial in the way that we plan to, what's the probability that we will produce the right answer as defined by those dichotomous declarations. So yeah, no, I'm honestly not quite sure what the answer is. I think people have thought about this. Other people have thought about this a bit more than I have, but it doesn't seem to have made much of an inroad. I gather that some trials are now being, I think I've seen this, that some trials are now being reported non-dichotomously per the Bayesian analysis. You know, you'll see reporting of posterior distributions, and maybe not just the probability that the effect is greater than this size, but some other probabilities. You can report many aspects of your posterior distribution. So, yeah, hopefully that will continue. But I agree that if you, yeah, and I kind of think maybe it's just a product of the fact that the world is still gripped by, we must control our type 1 and type 2 errors.
Yeah, thanks.
Thank you so much, Ian. Thanks, John. Unfortunately, we are unable to have any more Q&As. So yes, this is a recorded webinar, so the slides will become available. And let's see if it works. Yes, just some upcoming webinars for September, October. The speaker for September is known, but the date is still to be sorted. I think, Bernie, do you know what the topic will be in September?
Not at the moment, that's still finalized.
Okay, good. But in October, we have another interesting topic on the use of hospital data for research by Dr. Tanya Collier. So if you're interested, please register on the ACTA website. And finally, thank you so much. Thank you, John, for this presentation. You can never hear enough about p-values. And I think it's great to have had this webinar. If you also enjoy this webinar, I would recommend to fill in the survey and let us know how you thought it was. But also, if you have any topics that you would like to see in the future, feel free to fill them in as well. And maybe, Bernie, this link is sent to the attendees after the webinar. Is that correct?
Yes, it will. Yep.
Yeah. Okay, great. Yeah, I think this closes the webinar. So thank you all for staying after 3 and enjoy the rest of your afternoon. Thank you.
Thanks, Amina.